So my name is Jürgen Knoblich, and first of all, I wanted to express how much of an honor it is for me to be part of this extraordinary academy. So thanks a lot for accepting me into among this group of extraordinary scientists. So I am a developmental neurobiologist, and I'm interested in what I consider the most exquisite and the most complex and fascinating structure that exists in nature, which is the human brain. And the approach that we take is we want to understand the brain because we want to understand how it is actually formed during development. And for this, my lab a couple of years ago was able to generate a cell culture system where we take pluripotent stem cells that we can generate from any patient, healthy or diseased, through a process that was developed by Shinya Yamanaka, who is also part of this academy. And then we can take those cells through a cell culture procedure and grow them into cell cultures that resemble to quite some degree parts of a developing human brain. And as we can do this both from healthy and from patients suffering from neurodevelopmental diseases, we can recapitulate and investigate some of the defects that lead to these diseases. Now, my background is I was not always a neurobiologist. I grew up in southern Germany in a small town, a place where you need a catalyst to be confronted with science, and that catalyst for me was my chemistry teacher, and I can't emphasize enough the importance of education, even at a very young age, because without him, I would never have become a scientist, or more likely not. And he taught me two things. The first one was he sparked my fascination in chemistry, which was great for me, not so great for my parents, because I set up a chemistry lab and set up our garage in fire not only once. And the second thing he taught me was that he said, you know, the most fascinating area of chemistry right now is actually biochemistry. And so I went to the University of Tübingen to study biochemistry and slowly shifted laterally towards biology. What really determined the way I try to do science, and I try to also train the scientists in my group, was my time as a graduate student under the guidance of Christian Lehner in the vicinity of Christiane Nüsslein Volhard, where I worked with an organism called Drosophila melanogaster, or fruit flies, which is the best, one of the best understood animals of all. And what I wanted to know, I was fascinated by why the cells are actually dividing in a developing organism, and also why this stops at a certain point. And it was really great that during my PhD thesis, I found an answer to that. There's a particular gene that's downregulated, and that's why they stop. And what really fascinated me was that I could generate flies where this gene was not downregulated, and I was able to drive those cells to continue to divide further. And this moment of discovery is really what I find is very motivating and very important for any scientist. I then went on and I realized that there are certain cells in a fruit fly that divide into two cells that are not the same. They divide into two cells that look different in shape and in orientation. And in order to study that phenomenon, I went to the United States, to San Francisco, into the laboratory of Inang and Lily Jan, to study this process of asymmetric cell division, which is very important in stem cells, because stem cells generate both stem cells, but also cells that replace other cells in the body. And I wrote a proposal saying that, yeah, I want to find the mechanism of this as a postdoc. They accepted me, but I completely failed. There was I found a few molecules that are important for the process, but it was way too complicated. This was bad news for my postdoctoral supervisor, but it was great news for me because it allowed me to take that question on into my own laboratory. And within the first 10 years of having my laboratory, I think we and also other colleagues in the field were able to find a complete mechanism that actually guides this process, at least into in fruit flies. But then we were not completely satisfied with this. We wanted to show, does this also happen in higher animal brains? And we went into the mouse, but then in 2012, a postdoc arrived in my lab, and together we decided to take that one step further and to ask, can we analyze this process in the human brain? And for this, we generated this three-dimensional cell culture method based, of course, on the knowledge of many others in the field, but we decided to go one step further to actually use it to model certain neurodevelopmental disorders. And this is what I am totally fascinated by. Now we are trying to grow these three-dimensional cultures. They recapitulate the development of an actual human brain to a degree that is fascinating, sometimes even scary. The cells are arranged in a way that is very similar to a developing human brain. They fire, they interact with each other, and currently we're investigating on the neural circuit level to actually see what kind of functional processes are recapitulated. In this, we have been able to recapitulate certain disorders, like we have made models for epilepsy. We are working on autism, brain cancer. And I feel we are living in a time where in many areas of science, through the ever-advancing degree of technology that we have, we are really entering a new era. And this is particularly true in biology, where we are able to now bridge this gap between experiments that were for a long time done on animals and what we're actually interested in, which is human biology. And my vision for the next time is that by doing this, we can probably bridge the gap that exists between medicine and biology, both bringing biology closer to cures and to patients, but also helping to develop medicine in a way that is based on this knowledge and is experimentally approachable. And with this, I'm very happy to be here, and I consider it a big honor. Again, thank you.